Thank you, Claire, for that introduction, and thank you to Nanopore for another opportunity to present, to talk about something I love, which is how do we use long reads to improve clinical testing and find interesting variants that have positive effects on patients' lives. This is my standard disclaimer, and then my conflicts. And I'm going to start by apologizing. I'm going to reuse some introduction slides that you may have seen before. And the reason I'm going to do that is because I want to hold myself accountable for this timeline. I have thought for the past 2 years and continue to think that long reads will change clinical genetic testing. And I started saying this in 2023, so now it's 2025, which means we have 3 years left. And I think we are still on this timeline. As Claire mentioned, my lab is interested in doing this because we want to reduce the barriers for families, for individuals accessing comprehensive testing. We want to make it more equitable. And then we do think that this happens even if the cost of generating other types of data is $0. And that is because the value that you get with a technology such as long-read sequencing really surpasses, is much more important, easier to analyze than any other data type out there. Steve Jones and I last night were talking about this. You know, what really is the cost of 2 flow cells when you have an individual who's in the ICU on ECMO, right? It's really nothing. We think about this a lot in my lab with this diagnostic odyssey, this diagnostic workflow that patients are on. We go through this as clinicians with them, where we order a certain number of tests over time. Those tests may or may not be positive. Sometimes even when they're positive, we think that they may have missed something, and we're left asking, what is it that we have missed? This works for specific tests for rare diseases, but it also works for conditions such as cancer, where we send out multiple tests that may take days or weeks to come back, and often are either inconclusive or we really don't understand or agree with the result that we get. This is not really a linear process for individuals who are on this path. This is more of a complex game, if you will, such as snakes and ladders. I've learned how to say it properly, depending on what part of the world I'm in, where you might get one test or might get one referral that gets you a few steps forward. You might make a few shortcuts when you get a result back, but then something may happen that sets you back several steps. Our goal, of course, is to simplify this process. In a way, we want to make genetic testing boring, and I apologize for saying that, but we really just want it to be something that happens in the background, that everyone relies on, that everyone uses to get to really what we want, which is the treatment. But how do we start using this clinically? How do we show leadership at the hospital? How do we show the payers that that long reads are really what we should be using now? The people in this room, I think everybody kind of gets it, right? The talks today, the talks over the past few days have really shown the value of a single test, a single technology for evaluating individuals with rare diseases, with cancer, things like that. But how do we actually show folks that this is useful? We've done this over the years a couple of ways, just through examples of missing variant cases. So you saw Sebastian's talk earlier, using adaptive sampling to identify cases where you have one variant, but you don't find the second variant. So this is a classic one that I use. This is a kiddo who was born with a bleeding disorder. He had a panel, which was done on an exome backbone. It identified a variant in the gene FGA, which is the gene associated with this condition, but no second variant. So we used long-read sequencing without the parents to identify this 4 kb deletion that was missed by standard short-read sequencing. The family went on to use this for IVF, and every few years I get a photo of a new baby that was born after the clinic has tested the embryos. Another example is a kiddo that was referred to us who was known to have a glycogen storage disorder. He had extensive testing over the years. He had a panel-based test, he had an exome, he had a genome. The genome was done on a research basis and identified a variant in the gene associated with the condition that they thought was a translocation. That was done on a research basis, so then they had optical genome mapping done clinically, and that turned out to be negative. So this translocation that they thought they identified using short reads was actually not there. The question they had for us was, why was that? It turned out that the translocation was actually an ALU insertion, so the short reads couldn't fully resolve the ALU. The lab incorrectly interpreted it as a translocation, and long reads, of course, easily phased the entire gene. The pathogenic variant from the mom is on the maternal haplotype, but you don't see it here. But the paternally inherited ALU is the one that was missed. We've done this for a lot of cases. A lot of folks have done this, right? Solving missing variants. The thing that I've become more interested in, that our lab is more interested in, is applying this to larger cohorts of unsolved individuals. And we've started doing this in a couple of cases. And the one case I'm going to tell you about today is a group of individuals who have a condition called Canavan disease. Canavan is a pretty devastating neurodegenerative condition. It's one of those conditions where your child develops normally, and then at some point during development, you notice that they start losing milestones. And you ask why, you go see a pediatrician, they refer you to a neurologist, maybe eventually you make it to a biochemical geneticist. Most kids, or pretty much all kids who have this, will end up dying early, maybe before 10 years of age. And the thing about this condition is that there are some treatments that are being developed. So it's really important to get a molecular diagnosis for this condition. About a year ago, we were contacted by a team at Children's Hospital of Philadelphia, Adelaide Vanderveer and Carlos Dominguez Gonzalez. They had a cohort of 5 families, 6 individuals, who had a clinical diagnosis of Canavan disease, but either had an incomplete or partially complete molecular diagnosis. So the gene in this condition is called ASPA. I say ASPA. I actually don't know if that's correct, but that's what I say. And their families, it was 5 families with 6 affected individuals. In the process of working on this, we were actually contacted by a team in Australia, Katrina Bell, I heard that name earlier. They had identified 2 families with a similar story. They either had no variant identified in the gene or a single variant, and then no additional testing didn't identify a second pathogenic variant. Now, all of these cases had either exome sequencing, so short-read-based sequencing, or genome sequencing. And in all cases, they didn't find that second variant. For one of the cases, we got the short-read genome sequencing data, and I'm showing you that here in this IGV view. If you're not used to looking at short-read data in IGV, essentially the colored bars are reads that are either split or discordant, meaning they don't properly map to the area of interest. And I've zoomed in on this region because, spoiler alert, this is where the pathogenic variant's going to be. But the thing is, if you haven't looked at 100 different IGV or 100 different pathogenic variants in IGV, you may not catch this. And the other thing is that none of the short-read-based variant callers were able to identify the structural variant that's present here. And the giveaway for this is the little bump in coverage on that top. It's about 8 or 10 base pairs, and that's pretty characteristic of a transposable element insertion. When we did long-read whole genome sequencing on this case, we identified this 2,500 base pair insertion. This turns out to be an SVAE, which is a transposable element. These are pretty classically pathogenic by altering splicing. So we hypothesized that while typical splicing would produce a full-length transcript, when you have the SVAE, it's going to give you aberrant splicing. It's going to give you a shorter product that is then degraded. And we did confirm that using RNA sequencing. We do see that the colored reads you see down there on the bottom are evidence of aberrant splicing, and this is the pathogenic change. The thing about these variants is that they are amenable to therapies using ASOs, antisense oligonucleotides, so you can target that splice site and you can modulate that splicing effect. So it's really nice that with this data we have not only the presence of the SVA, but also we know the sequence and we can target it using an antisense oligonucleotide. And we're in the process of developing that. Why do we care for a rare disease like this? Well, we were really surprised when we went and asked, what's the frequency of this particular insertion? So these are the pathogenic variants in NOMAD for this gene. You can see that the most common one is present in about 0.003% of the population. And when you look for the SVAE, which is present in the short-read whole genome sequencing data in NOMAD, you can see that it's actually the most common pathogenic variant. So there are multiple children born every year in the world who are homozygous for this variant that are not identified by short-read sequencing. So this is a definite blind spot in our current use of the technology. I am basically out of time. I just wanted to give a quick update of how do we know that this variant is pathogenic. One way that we do that is we look at controlled databases, and one project we've been working on is sequencing samples from the 1,000 Genomes Project, and we have, I think, an exciting update at this point. So data from the 1,000 Genomes has been used for prioritizing and filtering short-read variants, so SNPs and indels, for example, using exome sequencing data. So one question we always had is how could we use this data to filter and prioritize structural variants? As of today, you can go to AWS and download data for 500 genomes from the 1,000 Genomes Project. The raw data is there, the POD5s, the align data is there, and the assemblies are there as well. And this is freely available to anyone online. What can you do with it? Well, you can filter structural variants. So if you take any one of us and you call structural variants genome-wide, you're going to find about 21,000, 25,000 structural variants. Using data from these 500 samples, you can filter this down to about 400 to 500 unique structural variants per person. This makes the filtering prioritization of these variants much easier. We have a tool called Needler, which is available online, and it can annotate variants for you, and it lets even people like me look at a file and say, oh, this individual has a family history of cancer. They have an AKT1 deletion that is rare, which probably explains the family history in this individual. So just to wrap up, the way I see the value in long-read sequencing is that today testing is stepwise. It takes weeks to years to, or months to years to complete, and sometimes testing really makes a difference in clinical decision-making. Where we're heading, where we're going to get to in 3 years is that the genetic testing will drive clinical decision-making because it will be available in hours or maybe a day or so, and it'll be kind of commonplace in the clinical setting. So I'll stop there and say thank you for your attention. Thank you again for the opportunity, and happy to take questions.